Welcome students. Last episode we discussed the protein synthesis in both prokaryotes and eukaryotes. In this episode we discuss the new topic genetic code and tRNA amino acylation. First of all, what is meant by genetic code? Synthesizing a protein with the help of the information in RNA is similar to translating a language to another. A 4-letter language is translated to 20-letter language during protein synthesis. There should be a specific relationship among the 4 bases of DNA and a sequence of 20 amino acids in the protein. What is meant by a genetic code? The genetic code can be defined as a set of certain rules using which the living cells translate the information encoded within the genetic material, that is DNA or mRNA sequences. The ribosomes are responsible to accomplish the process of translation. They link the amino acid in an mRNA specified, that is messenger RNA, order using tRNA, that is transfer RNA molecules to carry amino acids and to read the mRNA 3 nucleotides at a time. The complete set of relationships among amino acid and codon is said to be genetic code, which is often summarized as a table, that is genetic code table. It can be seen that many amino acids in the table by more than one codon. For example, there are 6 ways to write leucine in mRNA language. A codon is a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecule. A key point of the genetic code is its universal nature. This indicates that virtually all species with minor exceptions using the genetic code for protein synthesis. In other words, genetic code is defined as the nucleotide sequence of the base on DNA which is translated into a sequence of amino acid of the protein to be synthesized. The properties of genetic code are triplet code, non-ambiguous and universal, degenerate code, non-overlapping code, comma-less, start and stop codons, polarity. Triplet code. A codon or a code word is defined as a group of bases that specify an amino acid. There is strong evidence which proves that a sequence of 3 nucleotides codes for an amino acid in the protein. That is, the code is a triplet. The 4 bases of nucleotide, that is A, G, C and U, are used to produce 3 base codons. The 64 codons involves sense codon that specify amino acids. Hence, there are 64 codons for 20 amino acids. Since every codon for one amino acid means that there exists more than code for the same amino acid. Most of the amino acids being encoded by more than one codon. The genetic code is the set of rules by which information encoded in genetic material is translated into proteins by living cells. The genetic code, once thought to be identical in all forms of life, has been found to diverge slightly in certain organisms and in the mitochondria of some eukaryotes. The genetic code is the set of rules by which a linear sequence of nucleotides specify the nuclear sequence of the polypeptide. That is, they specify how the nucleotide sequence of an mRNA is translated into the amino acid sequence of a polypeptide. Thus, the relationship between the nucleotide sequence of the mRNA and the amino acid sequence of the polypeptide is the genetic code. The nucleotide sequence is read as triplets called codons. The principles of a genetic code. The genetic code consists of 64 different codons, each of which codes for one of the 20 amino acid. The genetic code is endowed with many characteristic properties which have actually been proved by definite experimental evidences. Comma-less code. No room for punctuation in between, which indicates that every codon is adjacent to the previous one without any nucleotides between them. The third property is non-overlapping code. The code is read sequentially in a group of 3 and a nucleotide which becomes a part of triplet never becomes part of the next triplet. For example, 5-UCU 3- codes for serine, 5-AUG 3- codes for methionine. The next property is polarity. Each triplet is read from 5- to 3- direction and the beginning base is 5- followed by the base in the middle, then the last base which is 3'. This implies that the codons have a fixed polarity, and if the codon is read in the reverse direction, the base sequence of the codon would reverse and would specify two different proteins. Degenerate code. Every amino acid except tryptophan UGG and methionine AUG is coded by various codons. That is, a few codons are synonyms, and this aspect is known as degeneracy of genetic code. For instance, UGA codes for tryptophan in yeast mitochondria. The next property is start and stop codons. Generally, AUG codon is the initiation or start codon. The polypeptide chain start either with eukaryotes methionine or prokaryotes N-formal methionine. On the other hand, UAG, UAA and UGA are called termination codons or stop codons. These are not read by any tRNA molecules and they never code for any amino acid. The next property is non-ambiguous and universal. The genetic code is non-ambiguous, which means a specific codon will only code for a particular amino acid. Also, the same genetic code is seen valid for all the organisms. That is, they are universal. Exceptions to the code. The genetic code is universal since similar codons are assigned to identical amino acids along with similar start and stop codon. Start and stop signals in the majority of genes in microorganisms and plants. However, a few exceptions have been discovered, and most of these include assigning one or two of the stop codons to an amino acid. Apart from this, both the codon GUG and AUG may code for methionine as a starting codon, although GUG is meant for valine. This breaks the properties of non-ambiguousness. Thus, it can be said that few codon often differs from the universal code or non-ambiguous code. Special codons. Chain initiation codons. The triplet AUG and GUG play double roles in E. coli. When they occur in between the two ends of cistern, they code for the amino acid methionine and valine, respectively in an intermediate position in the protein molecule. But when they occur immediately after the terminator codon, they act as a chain initiation signals or starter codon for the synthesis of polypeptide chain. Chain termination codons. The three triplets UAA, UAG, UGA do not code for any amino acid. When any one of them occurs immediately before the triplet AUG or GUG, it causes their release of the polypeptide chain from the ribosome. They are also called as stop codon. They are also called chain termination codons because these codons are used by the cells to signal the natural end of translation of a particular polypeptide chain. However, their inclusion in any mRNA results in the abrupt termination of the message at the point of their location, even though the polypeptide chain has not been completed. Sense codons. 61 codons which code for particular amino acids are termed as sense codon. Nonsense codons. Triplets UAA, UAG, UGA do not code for any amino acids. They were originally described as nonsense codons. However, the so-called nonsense codons have now been found to be of special sense. Their special sense codons perform the function of punctuating genetic messages like a full stop at the end of a sentence. Pattern of a genetic code. Amino acid with similar structure properties tend to have related codons. Aspartic acid codes GAU, GAC are similar to glutamic acid codons GAA, GAG. The difference being exhibited only in the third base towards the 3' end. Similarly, the codons for the aromatic amino acids phenylalanine UUU, UUC, tyrosine UAU, UAC and tryptophan UGG all being with uracil. All codons with uracil in the second position specify hydrophobic amino acid, that is isoleucine, leucine, methionine, phenylalanine, valine. All codes with the A in the second position specify the charged amino acid except arginine. All the acetic, aspartic acid, glutamic acid, and basic arginine and lysine amino acids have A or G as the second base. Reading frames and open frames. The mRNA sequence can be read by the ribosome in 3 possible reading frames. Usually only one reading frame codes for a functional protein, since the other 2 reading frames contain multiple termination codons. However, in some bacteriophage, overlapping genes occur which use different reading frames. An open reading frame ORF is a run of codons that starts with ATG and ends with a termination codon TGA, TAA or TAG. Coding regions of genes contain relatively long ORFs, unlike non-coding DNA where ORF are comparatively short. The presence of a long open reading frame in a DNA sequence therefore many indicate the presence of a coding region. Computer analysis of the ORF can be used to deduce the sequence of the encoded protein. The next one is amino acylation of tRNA. Transfer RNAs or tRNAs are molecules that act as a temporary carriers of amino acids, bringing the appropriate amino acids to the ribosome based on the messenger RNA nucleotide sequence. In this way, they act as a intermediates between nucleotide and amino acid sequence. tRNAs are ribonucleic acid and therefore capable of forming hydrogen bonds with mRNA. Additionally, they can also form ester linkages with amino acids and therefore can physically bring mRNA and amino acids together during the process of translation. They pair with the mRNA in a complementary and antiparallel manner, and each tRNA can base pair with a stretch of 3 nucleotides on mRNA. The sets of 3 nucleotides on the mRNA are called codons, and the corresponding sequence on the tRNA is called anticodon. Base pairing between the codon and anticodon brings specificity to the process of translation. On one end of the tRNA, an appropriate amino acid is attached to its 3'-hydroxyl group based on the anticodon, and the ribosome catalyzes the formation of a peptide bond between this amino acid and the elongation polypeptide chain. tRNAs are coded by number of genes and are usually short molecules between 70 to 90 nucleotides in length. The 2 most important parts of tRNA are its anticodon and the terminal 3'-hydroxyl group, which can form an ester linkage with an amino acid. However, there are other aspects to the tRNA structure such as the D-arm and the T-arm, which contributes to its high level of specificity and efficiency. Only 1 in 10,000 amino acids are incorrectly attached to the tRNA, which is a remarkable number given the chemical similarities between many amino acids. tRNA have a sugar phosphate backbone like all other cellular nucleic acid, and the orientation of the ribosugar gives rise to directionally in the molecule. One end of the RNA has a reactive phosphate group attached to the 5th carbon atom of ribose, while the other end of the free hydroxyl group on the 3rd carbon atom. This gives rise to the 5' and 3' end of the RNA, since all the other phosphate and hydroxyl group are involved in phosphodiester bond within the nucleic acid. The last 3 bases on the 3' end of the tRNA are always CCA 2 cytosines followed by 1 adenine bases. This stretch is part of a acceptor arm of the molecule, where an amino acid is covalently attached to the hydroxyl group on the ribosugar of the terminal adenine nucleotide. The acceptor arm also contains part of 5' end of the tRNA with a stretch of 7 to 9 nucleotides from opposite end of the molecule base pairing with each other. The anticodon loop, which pairs with the mRNA, determines which amino acid is attached to the acceptor stem. The anticodon loop is recognized by aminoacyl tRNA synthetase, the enzyme that chemically links a tRNA to an amino acid through a high energy bond. Aminoacyl tRNA synthetase reads the anticodon and also recognizes the D-arm located downstream from the 5' end of the tRNA. The D-arm is made up of double-stranded stem region formed by internal base pairing as well as loop structure of unpaired nucleotides. The D-arm is highly variable region and plays an important role in stabilizing the RNA's tertiary structure and also influence the kinetics and accuracy of translation at the ribosome. The other structure that influences the role of tRNA in translation is the T-arm. Similar to the D-arm, it contains a stretch of nucleotides that base pair with each other and a loop that is single-stranded. The The paired region is called the stem and mostly contains 5 base pairs. The loop contains modified bases and is also called the TPSI arm to specify the presence of thymidine, pseudouridine and cytidine residues. tRNA molecules are unusual in containing a high number of modified bases as well as containing thymidine, usually seen only in DNA. The T-arm is involved in the interaction of tRNA with the ribosome. The secondary structure of tRNA containing the acceptor region D and T arms and the anticodon loop is said to resemble a clover leaf. After the RNA folds into its tertiary structure, it is L-shaped with the acceptor stem and T arm forming an extended helix and the anticodon loop and D arm similarly making another extended helix. These two helix align perpendicularly to each other in a way that brings the D-arm and the T-arm into the close proximity, while the anticodon loop and the acceptor arm are positioned on opposite end of the molecules. Role of transfer RNA. Transfer RNA molecules play a key role in the process by delivering amino acids to the ribosome in an order specified by the mRNA sequence. This ensures that the amino acids are joined together in the correct order. Cells usually contain many species of tRNA, each of which binds specifically to one of the 20 amino acids. Consequently, there may be more than one tRNA for each amino acids. Transfer RNAs that binds the same amino acids are called isoacceptors. Before translation begins, amino acids become covalently linked to their tRNAs, which then recognize codon in the mRNA specifying that amino acid. The attachment of an amino acid to its tRNA is called aminoacylation or charging. The amino acid is covalently attached to the end of the acceptor arm of the tRNA, which always end with the base sequence 5-CCA3-. A bond forms between the carboxyl group of the amino acid and the 3-hydroxyl of the terminal adenine of the acceptor arm. Charging is catalyzed by enzymes called aminoacyl tRNA synthetases in a reaction requiring the hydrolysis of ATP. A separate enzyme exists for each amino acid and each enzyme can charge all the isoacceptors tRNAs for that amino acid. The aminoacyl tRNA synthetase recognizes both the appropriate amino acid and the corresponding tRNA. When the correct amino acid has been attached to tRNA, it recognizes the codon for that amino acid in the mRNA, allowing it to place the amino acid in the correct position, as specified by the sequence of the mRNA. This ensures that the amino acid sequence encoded by the mRNA is translated faithfully. Codon recognition takes place via the anticodon loop of the tRNA and specifically by 3 nucleotides in the loop known as anticodon, which binds to the codon by complementary base pairing. The entire codon-anticodon fitting is comparable to recognition of the 3-pin plug with the socket base. Both the pin and the socket are highly specific. The 4th bases present in DNA can combine as 64 codons. 3 codons act as signal for translation to stop and the remaining 61 encodes the 20 amino acid present in proteins. Consequently, most amino acids are represented by more than one codon. Activation of amino acid and attachment with the tRNA. Amino acid in cytoplasm occurs in inactive state. They are activated by gaining energy which comes from ATP. The reaction is brought about by binding of amino acid with ATR. The step is mediated by specific activating enzyme known as aminoacyl RNA synthetase. A high energy acyl bond is formed between the phosphate of ATP and the carboxyl group of amino acid with the formation of aminoacyl adenylate. The beta and gamma phosphates of ATP breaks away as inorganic pyrophosphate. The activated amino acid is transferred to its specific tRNA. A high energy ester bond is formed between the carboxyl group of amino acid and the 3-hydroxyl group of the terminal adenosine of the tRNA. The aminoacyl AMP enzyme complex reacts with the specific tRNA to form an aminoacyl tRNA complex. This is all about the genetic code and aminoacylation of tRNA. Thus we concluded this episode and we will discuss a new topic, translation proofreading inhibitor factors, in the next episode. Thank you.